Right, so we're going to start with DNA replication, so focusing on things that you read from chapter 6 and 13. We're going to start off by talking through the models for DNA replication, focusing on conservative, semi-conservative, and the dispersive models that led us to understand how DNA replication occurs in cells. We're going to do some comparisons between prokaryotes and eukaryotes so that you can kind of get a sense for the diversity and also the conservative nature of DNA replication across all organisms. And then we'll talk through some of the details of the proteins and molecules that are involved in the replication process. Then there are some examples at the end where we'll go into a little more detail about how mutations are fixed, how polymerase activity handles the ends of chromosomes and things like that. Okay, so last class we stopped talking about eukaryotic DNA organization, and we went through this picture that takes you from sort of the naked DNA molecule on this end with 2 nanomolar diameter, so over here, working through the first layer of DNA organization where DNA is wrapped around the histone proteins. So again, this is our DNA molecule. It's wrapped around histone proteins 2.5 times, and this combination of DNA and histone comprises the nucleosome. This is a relatively open euchromatic state for your chromatin. This is when we've got other proteins that can access maybe promoter regions that might be exposed on this section of the DNA that's not highly compacted. And so you might expect gene expression activity to be happening when the DNA is organized in this more loose structure. Moving on to the next major form that DNA takes in chromosome compaction is our chromatin fiber. This is also another common state that you'll find DNA in during interphase. This is a representation that looks more like the heterochromatin, so these nucleosomes are more tightly packed. And so our RNA polymerases, DNA polymerases are going to have a harder time accessing the DNA sequences that are wrapped around those nucleosomes. And then as we kind of zoom out even more, so this chromatid fiber is kind of zoomed in from this region of these chromatin fiber loops. We have additional proteins that are involved in compacting and condensing the chromatin so that it will fit in the nucleus of the cell. So the primary proteins involved here are condensins and cohesins. We also talked about the role of topoisomerases that can help manage the twisting of the DNA as it is compressed into smaller and smaller places. Then as we move to even higher orders of looping, so focusing on this kind of 700 nanometer version of chromatin versus the fully compacted chromosomes that you see during mitosis and meiosis, we can imagine these individual pieces of chromatin coiling and looping more and more tightly to the point where absolutely no gene expression is happening, and we're looking at a fully compacted chromosome that's been replicated and ready to go through cell division. So this is kind of where we're starting from. We also talked about some of the processes that manage how tightly packed chromatin is. So one of the mechanisms we talked about is acetylation. So again, thinking about this more open chromatin structure where we're looking at either this 11 nanometer version or the 30 nanometer version, our nucleosomes are more likely to be spaced out a little bit, but acetylation caused by histone acetyltransferase, or HAT, is going to increase the spacing between nucleosomes. In particular, our acetyl groups, which are carried on cofactors, are going to be transferred to our histone proteins on some of those lysine residues that are in the tails. And as a result of the transfer of the acetyl group, the positive charge of the histone protein gets neutralized so that the association between the histone and the DNA that's coiled around it becomes looser. And this allows those nucleosomes to move down and expose more of this DNA so that RNA polymerase, or if you're getting ready to do DNA replication, DNA polymerase can have access to that DNA sequence there. We talked about methylation and how it can have a context-dependent effect on the compaction of chromatin. The major way that we'll think about it in this class is through its role of condensing chromatin further. So where acetylation tends to open chromatin up, methylation can have context-dependent effects, but the example that I showed is a case where the addition of methyl groups causes the nucleosomes to become more packed together. In this particular situation, enzymes like RNA polymerase or DNA polymerase don't have access to the DNA sequence, and so they're going to be basically blocked from accessing and binding and carrying out processes like transcription and DNA replication. Very similar to the mechanism for histone acetyltransferase, this polycomb repressor complex 2 is going to require a methyl group that's donated by a methyl donating cofactor. That methyl group is added to different arginine and lysine residues that are found on the histone tails. And then the example that we walk through, and there's a screencast for this as well, is an example where the entire X chromosome might be condensed fully so that no gene expression is available from that particular chromosome. So in this particular case, I showed you an example of genetic mosaicism. This is a calico cat female, and there's a gene that codes for coat coloration that's carried on the sex chromosomes. And in female cats in particular that are heterozygous, so they're carrying a black allele and an orange allele for the color gene. The gene that gets expressed is dependent on which of the two X chromosomes is inactivated. So if you were to zoom in into the nuclei of the cells that have this orange coloration, you'd see a case where the X chromosome carrying the black allele is condensed and not expressing anything, and only the orange allele is expressed. And if you zoomed in to the black patches of fur, you'd see the opposite case. So the X chromosome carrying the orange allele is not expressing anything, and the X chromosome carrying the black allele is expressing, leading to black coloration in the cat coloration. So that is where we're coming from. One of the things that we're going to focus on moving forward with material today comes back to central ideas with the central dogma of gene expression. So I've shown you a similar sort of image so far this semester. This one provides a little bit more detail on some of the mechanisms and the icons that you'll see me use multiple times. So I wanted to kind of get them familiarized at the beginning of this kind of material. But basically, this is a representation of information flow from DNA to expression. So one of the reasons that the central dogma is an organizing theme in genetics is because this process of DNA replication, transcription, translation, and expression is universal across all organisms. So all genetic material needs to be capable of replicating, being transmitted to the next generation of cells or individuals, and it needs to be able to express some information so that you can have form, function, processes, any other kind of trait that you would be interested in measuring in an organism. So today we're going to focus on the DNA replication aspect of the central dogma, where we're going to be able to take information in the DNA double helix and go through the process of replication so that it can be transmitted to the next generation. Coming back to the characteristics of genetic material that we've been talking about so far. So we've covered information storage. We've talked a little bit about transmission of genetic information. Today we're really focusing on point 3, where we're interested in understanding how genetic information is passed from parent to offspring or mother cell to daughter cell. You can think about this kind of at 2 different levels there. But the key thing is coming up with a way to account for the need of a template in order to make an accurate copy of this genetic material and pass it down. So at the time of Watson and Crick's model that was put forward based on X-ray diffraction data, Chargaff's rule, and basic understanding of the characteristics of nucleotides, there were sort of 3 models that were put forward to demonstrate how DNA is replicated. These 3 models are conservative model, semi-conservative, and dispersive model of DNA replication. So these three things are defined for you on the screen. I'm going to show you animations of how these conservative, semi-conservative, and dispersive models are expected to have worked at the time. Okay, so we have our conservative replication model up here. We're starting off with a parental version of the DNA double helix. So this is shown in orange in each of these examples. With conservative replication, the expectation is that the two strands of the double helix are going to remain together, or they both come apart and pair again in the same way after replication is complete. And so what this looks like is an exact copy of both strands of the double helix is made, and when you're done with DNA replication, you have two copies, one entirely old strand, which is this one here that we started with, and one entirely new strand of DNA double helix. The next kind of model for replication is the semi-conservative replication model. So again, we're starting off with our parent DNA double helix shown in orange. With the semi-conservative replication model, you can see from our starting point, we're ending up with two molecules that have each one parent strand, which is shown in orange, and one nascent strand or newly replicated strand, which is shown in blue. So with the semi-conservative replication model, instead of using the entire molecule as a template, we're taking this molecule, unwinding it, and using one strand to as a template for the two replicated molecules that are produced. So in particular, this strand was used as a template to produce that molecule, and this strand was used as a template to produce that molecule. And then our third replication model is the dispersive replication model, or hypothesis that could be put forward. This is the idea that this parent DNA molecule is essentially broken up into multiple pieces and sort of stitched back together. So the first step in this procedure is that these parent strands are cleaved into double-stranded DNA fragments and then reassembled with interspersed, newly copied or nascent black DNA segments. So these orange copies are going to be used to make a replicate version, so something that looks like this. So the black DNA double-stranded fragment is a copy of this orange one here. And after all of those copies have been made, these disjointed pieces of DNA are then put back together to form our two strands of DNA that are produced. So out of the three models, this one is definitely the most chaotic. There's a lot of things that could potentially go wrong if you're going to break apart and try and reassemble pieces of double-stranded DNA segments. It's pretty easy to see how you might end up with rearrangements that aren't intended. But in each of these cases, we're ending up with two DNA molecules. In one case, the new DNA molecule is an entire copy of the old DNA molecule, which is the conservative situation. The semi-conservative replication model, we end up with two new molecules that each contain half new and half old DNA strands. And then the dispersive replication model gives us two hybrid DNA molecules as well, but instead of the strands being continuous, we have these sections that are stuck back together. Okay, so the key experiment, or one of the key experiments that was able to differentiate between those three models of DNA replication was carried out by Messelsons and Stahl. In this work, they were focusing again on a model organism instead of the virulent strains that we talked about in respect to figuring out the genetic information. Now we're using E. coli, so a different bacterial species that is relatively simple, fast, and easy to grow in a laboratory setting. So these bacteria cells are being grown in a special type of medium or a special type of growing solution. So medium is basically just a nutrient-rich environment for these bacteria to grow. And in that media are all of the nutrients that bacteria need to replicate their DNA, to go through cell division, and to continue to expand the population size. And the idea here is the specific attributes of the nutrients that are found inside this nutrient broth are going to have an influence on the chemical structures that the bacteria are building as they build their DNA molecules. And this is particularly relevant for this experiment because the type of nitrogen that was included in this growth medium is a special kind that's not normally found in nature. So N15 is a heavy form of nitrogen that literally makes the molecules that use that type of nitrogen heavier in comparison to the molecules that are made using normal nitrogen, or N14. So as this strain of E. coli grows and divides in this N15 labeled media, every time DNA replication happens, nitrogen-containing molecules are going to be incorporated into that DNA replication process. So in particular, if you remember the basic structure of a nucleotide, you have your phosphate group bound to your pentose sugar, which is bound to a nitrogenous base, right? And these nitrogenous bases are nitrogenous because they have nitrogen in them. And so every time the DNA replication process happens and new nucleotides are needed, those nucleotides are going to have this heavy form of nitrogen in them. So naturally occurring nucleotides have N14. They're going to have a lower molecular weight compared to our nucleotides that have this N15 version. They're going to have a higher molecular weight, literally weighing more, making the DNA heavier. And the reason that matters is if you were to take the sample from this flask of bacteria growing in N15 labeled media and centrifuge it, the weight of those molecules will cause the DNA to go to the bottom of the tube. So here you see the band that corresponds to DNA extracted from bacteria growing in N15 media. And because both strands of that DNA double helix have N15 incorporated into the bases, we have a settling out of a heavy band of that particular DNA. Okay. So the next step in this process was to take these bacteria that were grown in the N15 labeled media and transfer them to clean N14 media. So this is the type of media that's going to have the lighter version of nitrogen, and the bacteria are allowed to go through one round of replication. So thinking about our conservative, semi-conservative, and dispersive models of replication, for every molecule of DNA in this particular flask in generation 1, we're going to have one exact copy of that. After that round of replication was completed, DNA was extracted from those bacterial cells and then subjected to centrifugation again. So where we started in generation 0 with all of the DNA containing molecules having this heavy form of nitrogen, in generation 1, we see this mix. There's evidence that there's some kind of hybrid form of DNA. So half of it has the parent version of nitrogen coming from this original generation 0 in the pink N15 media, and half of that DNA is going to come from the new round of replication. So these are the new DNA molecules that are formed, and because that new DNA replication occurred in a place where only N14 is available, N14 is incorporated into that side of the DNA molecule. So we have this movement of the band from the end of the tube to the middle of the tube. They carried this experiment forward for another few generations. And we can use the information coming up from this generation, generation 1, and the following generations to start ruling out conservative, dispersive, and the semi-conservative models of DNA replication. So at this point, I would typically pause and say, which of the 3 DNA replication models that we've discussed can you rule out at this point? And it's so sad doing screencasts with no one else in the room because I wait for nothing. Okay, anyway, So conservative replication can be ruled out. Why is this the case? So if we think back to our DNA molecules, I'm not going to draw a double helix because it's going to look terrible, but we can have our parent version of the double helix. And with the conservative model of replication, we have an exact copy. And I don't know if I have a different color. Let me see. I don't think I do. That's too bad. We have the parent version and an entirely new version of the DNA double helix over here. So none of this information, none of the molecule that comprises the parent chromosome is going to be incorporated into the new DNA molecule. If this was the case, if we did have the parent DNA molecule that stayed separate from the new DNA molecule, we would expect to have two bands in this outcome. So a case where N15 still exists and our N14 still exists. Separately. But because we have a hybrid, this suggests that the new DNA molecules have to have some hybrid component of them. One strand is N15, the other strand is N14, or perhaps there's this dispersive model where you have N14 followed by N15, followed by N14, followed by N15, etc. So we can't rule out the dispersive model yet. Okay. So moving on to generation 2, doing the same set of experiments. So these cells from generation 1 are allowed to replicate one time to give generation 2, so one new round of DNA replication. The DNA is extracted from those cells and centrifuged again, so we're looking at this one now. And now we start to see some separation of these bands. So we have our original hybrid kind of form of DNA where there's some molecules that have both N15 and N14 in them. But then there's this new set of molecules that have N14 and N14. Either stacks one after another, or one strand is N14 and the other strand is N14. Carrying this forward again, we have a third replication, so looking at generation 3, the DNA is centrifuged once more, and we see that this quantity of molecules that has N14 only is starting to increase relative to the hybrid versions of the molecules. So this initial component where we've got some DNA molecules that have N15 incorporated into their DNA structure, that quantity doesn't change over time, but we see more and more of these newer molecules, these newer DNA structures, new chromosomes that only have N14 incorporated into their bases. Okay, so from here, from this standpoint, we can rule out conservative DNA replication. From this standpoint, thinking about generation 2 and generation 3 together, we can start to think about whether dispersive or semi-conservative DNA replication is more likely. All right, so to help kind of illustrate this, I have the same sort of experiment broken down in a slightly different way on this next slide. And so this would be one of those in-class activities that I was planning to have you work on. And what I want you to do is take 5 minutes or so to make predictions about what you would expect to see using the tubes that are kind of drawn here. If you have conservative DNA replication versus dispersive replication. If this is the expectation that the current results that were observed support semi-conservative replication. So if you wanted to like literally draw this out, you could have, you know, your little test tubes and predict what you'd see. Where would you see bands in each of these places? So take 5 minutes, pause the video, see if you can work that out, and demonstrate the type of evidence that you need to support those other hypotheses. Okay. So we're going to move to the answer now. So with our conservative model of DNA replication, if we were going to draw out what you would expect to see if this conservative model is the correct DNA replication model, you'd have something that looks like this. So we have our test tube after centrifugation, and sort of like I showed you on the previous slide, we would expect to see this N15 version of DNA persist because that parent version of DNA always stays together, and then an exact copy with N14 incorporated into the two strands of the double helix, always being separate from the old version, the old parental version of the DNA. And this is not what we see in any of the experiments that are outlined above. And so as a result of that not being observed, we can rule out conservative replication of DNA. Okay, so let's think through the dispersive case now. So we might expect to see a similar sort of result to what we actually see in generation 1 if dispersive replication is happening. So again, this is the idea that if you have your parent and your newly replicated sections of DNA interspersed in these double-stranded chunks, you might have something like this, where all of the bases here have N15, all of the bases here have N14, and so on and so forth. However, when you get to generation 2, there's no reason to expect this to not continue to happen, right? So generation 2 should also have a total kind of global hybrid version of the DNA molecules. They should all have some DNA that has N15 and N14 in them if the dispersive model is the one that's happening. But instead, we'd see that there's a separation of these bands in the actual results, different from what you might expect to see. So because these DNA molecules are starting to separate out, where one of these strands is used as a template, and as a result produces DNA molecules that are entirely composed of N14, we can rule out dispersive DNA replication as a model for DNA replication. Okay. So I know that it's kind of confusing to hear N14 and N15 over and over again, but if we think kind of back to our replication type in the cartoon version, what we're really doing is we're saying this version of replication can't happen because we always have some version of hybrid molecule. So conservative is ruled out. Both conservative, semi-conservative, and dispersive replication models have some type of hybrid molecule that's produced, but because the dispersive replication model always expects some interspersing of maybe this is N15 and this is N14 containing bases, we always expect to see that kind of hybrid band in the middle of that test tube. Something like that. Whereas over here, over time, in the next round, in semi-conservative replication, and the next round of DNA replication, this strand is going to become a template, and this strand is going to become a template, and you'll still have an N15, N14 molecule that incorporates, sorry, my colors are backwards. Let me try that again. If this is our N15 containing double helix, and this is the output of generation 1 DNA replication, when it comes time to look at generation 2, when this orange strand is used as a template, it's going to produce a molecule that has N15 and N14 in it, and this new strand in blue, which has N14 in it, is going to replicate in an N14 environment and produce double helices, new chromosomes that have that 14-14 band combination, which, if you think back to your test tube, gives you the 2 bands. So this is our hybrid band, and this is our new lighter version of the DNA molecule. Okay. So we can move forward with the semi-conservative DNA replication model. And if you think back to what you remember from the cell cycle and when genes are expressed, when mitosis happens, et cetera, you know that there are specific phases of the cell life cycle. We can divide these into G1 phase, S phase, G2 phase, and M phase. G1 phase is what our cells are in during interphase. This is typically before chromosome duplication or before DNA replication happens. Then, in preparation for moving towards M phase, when cells are actually dividing, DNA synthesis occurs during S phase. So S phase is for DNA synthesis, if you want an easy way to remember it. This is where we have DNA replication happening and where we go from chromosome structures that are a single chromatid to a duplicated structure held together at the centromere. So this DNA duplication process gives us those typical kind of X-shaped chromosomes that you're used to seeing. Then the cell will go back into G2 phase, where it's continuing to prepare for cell division, and then M phase happens. So this can be mitosis or meiosis. Depending on whether you're looking at a somatic cell or a germline cell. So what we're talking about today happens during this S phase. There are 3 phases of DNA replication, and we'll start off with the initiation phase. Initiation is a little bit different between bacteria or prokaryotes and our animal cells, eukaryotes, if you will. And the major reason for this difference comes down to the amount of genetic material that's present in these different types of cells. So with bacteria, you have relatively small chromosomes, relatively little genetic information, whereas in eukaryotes, we have much larger genomes. Multiple chromosomes, the chromosomes themselves are much longer, many more nucleotides are used to build those structures. And as a result, it takes longer and is less efficient to replicate the entire genome of a eukaryotic cell as opposed to bacteria. And that difference in genome size and complexity leads to the initiation differences. So with bacteria, we have a single point of origin, which is what's shown over here in this circular chromosome. So bacterial chromosomes are circular. There's a single origin of replication, and this has a particular DNA sequence. So at this particular site that's kind of highlighted by purple, we have a bunch of ATs repeated over and over and over again, and that DNA sequence is recognized by the DNA A protein. DNA A is an initiator protein that binds to that AT repeat region, and it's going to allow initiation to start, the replication to start. Eukaryotes have a key difference here. There's many points of origin per chromosome instead of a single point of origin. And sort of the analog to DNA A is this pre-replication complex. So this is going to control the timing of DNA replication by marking the origins at the start of each G1 phase of the cell cycle. So kind of going back to this, those pre-initiation complexes are going to start marking the DNA at the beginning of G1. The rest of G1 goes on and allows genes to express, the cells to grow, to express different genes, carry out different functions. And then by the time you get to the S phase, those marked origins of replication are going to be the starting point for DNA replication to occur. Okay. So as opposed to our bacterial chromosome, which is circular and has a single point of origin, in our eukaryotes, we have long linear chromosomes and multiple origins of replication that are marked by those pre-replication complexes. I think I called them pre-initiation complexes, but pre-replication is more appropriate. So our pre-replication complexes are basically going to mark these origins of replication so that during S phase, the DNA polymerases and all of the other proteins are going to be able to recognize those sites and start replicating DNA. So that process, kind of at a broad scale, involves unwinding the DNA double helix so that each of these strands can be used as a template for the new DNA strand, which is shown in that pinkish color. And at each of those points of origin, you have expansion of the DNA molecule kind of out from the center until you get connection points between those individual origins of replication. And ultimately, this allows, as this continues in both directions, those two chromosomes to separate, those two copies of the same chromosome to separate. Okay. After initiation has kind of either identified places to start or started the replication process, the next phase is elongation. This is pretty similar in prokaryotes and eukaryotes, and a lot of what I'm going to show you today has been really clearly fleshed out in bacteria, and we're still learning about some of the mechanisms that are used in eukaryotes, but there's quite a lot of similarity. There are a couple of key things that are true for both organisms. So in both prokaryotes and eukaryotes, the key molecule for catalyzing the polymerization reaction, or the chain building of DNA, is called DNA polymerase. This is what's going to facilitate joining new nucleotides to the preceding nucleotide and form those phosphodiester bonds. It's always true that DNA strands are growing in the 5' to 3' direction, and this has everything to do with that kind of receiving end of the nucleotide. So phosphate to your sugar. The 3' end has that OH group, which is going to receive new nucleotides at this location. And elongation, DNA replication in general, is bidirectional, which means that it proceeds on both template strands of the double helix at the same time. And you might have heard these terms before, leading and lagging strands. This is going to become relevant as we talk about differences in how the DNA nascent strand, the newly synthesized strand, is being put together on the two different sides of the double helix. So you've seen pictures that look like this before already this class, but this is kind of to remind you how chain elongation happens with DNA. So DNA elongation occurs in the 5' to 3' direction. And so what this literally means is if you look at the growing chain of nucleotides, which is what's shown here, the 5' end of the carbon of the pentose sugar is attached to the phosphate group, and the 3' end of the growing chain is attached to this hydroxyl group. When a new nucleotide is going to be added to the growing chain, the 5' end is going to be attached to the 3' end. This particular image is showing you a different kind of precursor nucleotide than what I've shown you in the past. So this is specifically a nucleoside triphosphate, referring to the 3 phosphate groups that are attached to this nucleoside. 2 of these phosphate groups are cleaved off, and the energy that's released through the cleaving of that phosphate bond allows the phosphodiester bond to form between this phosphate group and the 2-deoxyribose sugar that's shown in this picture. So ultimately what you get is the 2 nucleotides that were originally part of the DNA chain and this new nucleotide that's been added, producing, like we've seen before, a water molecule, as well as these 2 phosphate groups. Okay. So our next sort of step with understanding DNA replication in the elongation phase focuses on several enzymes that are all responsible for unwinding and exposing the DNA double helix template so that replication can happen. A good way to study this information is to use flashcards. So flashcards aren't great for every type of kind of study technique, but for when you're trying to remember what specific things will do in a series of events, these terms will be ones that you'll want to have ready definitions for, and so flashcards can be really helpful with that. Okay. So we're going to step through each of those enzymes and proteins and structures that are indicated over on the left-hand side of the screen. And I'm going to reference this sort of trombone-shaped molecule model of DNA replication. So a couple of things to point out about this model of replication. We have our parent strands, which are shown in blue. So this is always going to be the parent molecule. And the newly synthesized strands are shown in yellow. Okay. Our DNA polymerase, which is this big kind of blue molecule here, is moving in this direction. So we're basically coming from the bottom of the screen down here and moving up to the top of the screen as you're thinking about the direction of how this DNA replication process is happening. So there comes a point right here where we're going to talk about helicase first, where the kind of several protein components that are responsible for DNA replication eventually meets this bound up DNA double helix. And so we're kind of moving the helicase and the double helix following this direction towards the top of the slide. All right, so helicases first, what we'll talk about first. Helicase is this protein that binds at the replication origin. So in eukaryotes, this is going to bind following pre-replication complex proteins. In bacteria, this is going to follow DNA A. Our helicase is this green component right here, and it's going to start unwinding and denaturing the DNA. In particular, denaturing refers to breaking hydrogen bonds that are formed between the base pairs. So literally kind of unzipping this DNA molecule in this direction. As that happens, the parent strands are now single-stranded. And in order to prevent them from kind of snapping back to form that base pair structure that they want to form and reforming those hydrogen bonds that were just broken by helicase, these little white single-strand binding proteins, which are labeled right here, line the exposed base side of the single strand of DNA. They're basically protecting it from reannealing and reforming that DNA double helix. And as the DNA polymerase moves along the template strands, these single strand binding proteins are removed. In order for any DNA replication to start adding new nucleotides, we need to have a modification made by primase. Primase is indicated by this yellow protein that follows right after the helicase. And you can see that it's bumped off some of those single-strand binding proteins so that it can add an RNA primer. This RNA primer is important because it provides the 3' OH group that is available for the next nucleotide to be added. You might see NT throughout the PowerPoint presentation. NT refers to nucleotide. So primase is a specific type of RNA polymerase that's going to synthesize this RNA primer following the typical base pairing rules. In eukaryotes, it has a slightly different name, so you might see it referred to as polymerase alpha. And once again, the primary role of primase or polymerase alpha is to provide the starting point for new nucleotides to be added. Our next key player is DNA polymerase 3 in particular. So we're looking at this big blue protein here in the center. DNA polymerase 3 is going to add nucleotides in a very specific manner. So like we've said, they're only going to be added to the 3' end of an existing strand. So at the very beginning, this is going to be added to an RNA primer. And after that first DNA nucleotide has been added, the rest of the DNA nucleotides will continue to be added to that existing strand. DNA polymerase is a big, complex protein. It has multiple components, and it's able to catalyze its own reaction, which means that it's able to actually form those phosphodiester bonds that need to exist between different nucleotides as they're added one next to another. So this large enzyme complex is referred to as a holoenzyme. It's a combination of multiple subunits and cofactors that are going to be able to carry out this activity of adding new nucleotides one after another. So the holoenzyme, refers to this entire structure that's shown in green, purple, whatever that color is, and orange or gold or whatever. There's a key component that helps stabilize the DNA polymerase as it's moving down the DNA template, and that is this DNA sliding clamp. This is basically what it sounds like. It clamps to the DNA template and allows the polymerase to move along without skipping around. It's going to start from the RNA primer, and it's going to proceed with DNA replication on both the leading and the lagging strands of DNA simultaneously. So looking back at this model of DNA replication, this would be the leading strand, and this one would be the lagging strand. And there's a key difference between these two sides that tells me that this is the case. And if you guessed that it was the fact that these segments of DNA are in sections called Okazaki fragments, that is the key to telling whether or not this is the lagging strand versus the leading strand. So these DNA sections are synthesized in shorter fragments, whereas on this side, there's one continuous new strand of DNA. So that's how you tell the difference between the leading and the lagging strand. Okay. So I'll say this a lot because it's important to remember and really easy to get confused. These new DNA nucleotides, DNTPs, are going to be added to the 3' end of the growing strand. And this is a little bit confusing to think about because the new strand is growing in a 5' to 3' direction, but as the DNA polymerase is moving down the template strand, it's actually moving down the strand in the 3' to 5' direction. So a little bit of a distinction there to keep straight, and we'll come back to that a bunch of times. And I think I might post kind of a drawing of how all of this works a little bit later. Okay. Moving right along, we have our differences between leading strand and lagging strand synthesis. So I mentioned in the previous slide that we have fragments of new DNA chains that are being synthesized on the lagging strand. So this lagging strand is referring to that yellow, newly synthesized DNA. So Okazaki fragments are these short lagging strand fragments. They require an RNA primer to get started, just like with the leading strand, and they proceed in the 5' to 3' direction as well. So this means that they're being synthesized in the 5' to 3' direction. So again, in this picture, our RNA primer is shown in red as our starting point, and the new DNA is shown in yellow. All right. So these smaller fragments, Okazaki fragments, are present in both prokaryotes and eukaryotes. But one of the big differences between prokaryotes and eukaryotes is the overall size of Okazaki fragments. In bacteria, we have Okazaki fragments that are about 1,200 nucleotides long. And they're much, much smaller in eukaryotes. So there's quite a range in terms of how long Okazaki fragments are on average, but you'll see anything between 100 and 300 nucleotides long in eukaryotes. And the reason for this has a lot to do with what we talked about last class, with the overall organization of DNA in the cell. So why would it be the case? Why would you expect to see different size Okazaki fragments in prokaryotes and eukaryotes? So take a pause, think through the answer. If you chose the answer nucleosomes, you would be correct. So the spacing of histones, or the nucleosomes, are going to have a direct relationship or a direct impact on the size of the Okazaki fragments. In prokaryotic cells, which is shown here, the primary way that DNA is organized and compressed into the cell, the nucleoid region, is through supercoiling. So that process of overwinding or underwinding the DNA so that it backlashes on itself. In eukaryotes, DNA organization involves the role of histones, and those histones, which form nucleosomes, are going to be regularly spaced, for the most part, along the DNA. And as you'll recall from what we talked about with histone acetyltransferase and methyltransferase, the proteins, such as DNA polymerase, are going to be able to access the DNA in these regions between those nucleosomes. So, as these Okazaki fragments are being formed, and also as the leading strand is being synthesized, we have to do something about nucleosomes in order to allow DNA replication proteins to make their way up and down the template strands. So in this picture, we have our replication fork again. So we've rotated it. So instead of going up the screen like we've been looking at in the previous diagrams, now we're going to the left. So DNA polymerase isn't shown, but you could imagine it kind of, moving along this section of DNA, and new nucleotides are being added to this end. So as this replication fork continues to push apart the DNA double helix, we have to do something with these nucleosomes and the histone proteins in particular. So these are octamers, remember. One of the things that immediately happens is the DNA unwraps from around those histone proteins, and then the histone proteins themselves become less stable. So those different subunits are going to separate, and parts of the old histones are going to be reused as the DNA polymerase moves past sections of DNA that's already been replicated. So you can see that happening here. This light blue color is being reused as a subunit on this newly replicated strand. Whereas at the same time as DNA replication is happening, new histone protein subunits are also being synthesized, and those are going to be incorporated so that we have enough nucleosome formation given the amount of DNA that's being produced. And so you can see different components of these old histone proteins being reused in other parts of this diagram. So while DNA replication is happening, we still have other processes like transcription and translation, primarily of the proteins that are involved in the DNA replication process. And one of those are those histone proteins that are important for organizing and storing this DNA. Because if you think about it, when you're taking a genome and doubling it, essentially, that ability to organize and condense the DNA into the cell is going to become even more important. Because not only do you have 6 linear feet of DNA in a human nucleus, now you have 12, and that has to be compressed even more. Okay. So recapping a little bit, our Okazaki fragments in eukaryotes are typically much smaller. So I have 100 to 150 nucleotides. I've seen 100 to 300 nucleotides long. Generally, much smaller than the 1200 nucleotide long fragments that are seen in prokaryotes. In general, the formation and synthesis of DNA on the lagging strand is more complex in eukaryotes versus prokaryotes. And this is primarily because there are just more Okazaki fragments. Because even though those fragments are smaller, the eukaryotic genomes are typically much, much larger. So not only do you have more DNA, you have more fragments to stick back together in order to complete a full version of the DNA double helix replication. These Okazaki fragments, given their number and given their size, contribute to complexity because for every Okazaki fragment, you have this initiation point and then an RNA primer that's followed by DNA. And we know that when DNA replication is complete, there's no RNA nucleotides incorporated into the actual double helix structure. And so for every one of these RNA primers that's present, something has to happen to remove those RNA primers and then fill the gaps that's left behind. And so this is carried out by additional enzymes that we're going to talk about. Polymerase delta is the specific component of polymerase 3, DNA polymerase 3, that big sort of rabbit-eared shaped protein, the hollow enzyme that we talked about with the sliding clamp. Looks like a sad face. One of these subunits of that big DNA polymerase 3 is called polymerase delta. But this enzyme itself can only synthesize new DNA. It can't take away and fill the gaps that are produced by these RNA primers once they're removed. That process is carried out by polymerase 1. So polymerase 3 is our major polymerase replication engine. Polymerase 1, this purple enzyme, is going to be responsible for removing the RNA primers and then filling in the gaps that are left behind. And specifically, it does this with its 5' to 3' exonuclease activity. And I have that diagrammed in a little bit. We'll come to that. The last little bit of processing that needs to happen on the lagging strand is sealing the space, the gap between the last nucleotide on one Okazaki fragment and the first nucleotide on the next Okazaki fragment. So this ligase protein, which is shown in yellow, is basically going to heal the gap between those two different Okazaki fragments. Then we also have topoisomerase or DNA gyrase in prokaryotic organisms. We've talked a little bit about topoisomerases. You can recall how many DNA strands are nicked or broken by this type of topoisomerase based on the number that follows it. So DNA gyrase is going to be able to induce and also relax supercoiling ahead of this DNA double helix, ahead of this helicase, allowing the DNA double helix to open and accommodate the activity of the DNA polymerase. All right, so the DNA replication video that I would normally show in class is actually linked on the Canvas site. So I'm going to take a pause so that I can take a drink and come back to this a little bit more refreshed, but take a stop and look through that video. I think it's 2 minutes long, and you can kind of see how all these processes work together.